Perfect. All right, we're going to get started and we'll let people keep joining us. The next few minutes people will roll in, but happy November, everybody. I'm really happy to welcome you all to a webinar on early detection and prevention of cancer. And we are honored to have two oncologists from the Dana-Farber Cancer Institute, Dr. Betsy O'Donnell, who's the director of the Multi-Cancer Early Detection Clinic, and Dr. Sapna Singhal, who is one of her co-leads and also the director of cancer genetics and prevention at Dana-Farber. So without further ado, I'm going to introduce both of you, and I want to remind everyone to please feel free to submit questions in the chat or the Q&A, and we'll get to those at the end. We're going to save some time for everybody's questions. So thank you, Betsy and Sapna.
Thank you. So it's our pleasure to be here. I'm going to start us off, and then Dr. O'Donnell is going to take over for the second half of the presentation. And we're really excited that Dr. Lewis asked us to sort of present this new area that Dana-Farber and across Harvard and our sort of medical cancer care community has gotten really excited about over the last few years. The whole idea, and Betsy will go into this in more detail, is we've known for a long time that outcomes for cancer are completely related to the stage of diagnosis of cancer. So picking up cancer early really leads to the best outcomes. And all of you know this already intuitively. The other thing is that if we can actually even jumpstart that a little bit more and start taking care of people about their cancer risk earlier before a cancer diagnosis, well, that's even much better, right? Because then we're looking at prevention or interception, as we're calling it. And Dana-Farber and the Harvard Cancer Center has been really at the forefront of this over the last 5 years. And we've decided that part of our strategic plan for the Cancer Center and nationally in this field is that we really need to be really putting a sharp lens and a sharp focus on this area of early detection of cancer and interception. Prevention used to be used all the time, and it's kind of gotten out of fashion because people felt it was kind of judgy, meaning prevention is like, telling people to just, you need to lose weight or you need to stop smoking. It was sort of felt to be very like sort of paternalistic. Interception is like the football analogy. We're going to intercept, we're going to catch the ball, and we're going to actually prevent that whole progression of going on to cancer. So lo and behold, about 2 years ago, we launched at Dana-Farber the Centers for Early Detection and Interception. And this is our website. If you ever want to look more deeply into this, please visit us on the web. And Betsy and I are talking today, but our other partner in this is Irene Gabriel, who has been really a maverick in treating early myeloma precursors before people get multiple myeloma. So we're not going to talk a lot about that today, but that's a really integral part of the program as well. And next slide. And what we decided is that the mission of these centers is, as I said, to stop cancer before it starts. And the other thing we say now is that Dana-Farber right now is a place to go for if you have cancer. We also want Dana-Farber to be the place to go if you don't want to get cancer. And what we decided very sort of strategically is we're going to focus these centers on caring for patients who are at elevated risk of developing cancer. And we'll talk about a little bit about what we mean by that. But we're not talking about general population risk for average risk of cancer. So, for your colon cancer screening or your routine breast mammography, your PSA testing, your primary care physician is still the best place to kind of manage all that. But there's this middle ground between getting cancer, people with cancer, versus the general population of people who have an elevated risk. And that's what these centers are catering to. Next slide. And what Dana-Farber has done is made a massive investment in this, in these centers. So when we got approval about 2 years ago, these are all the people that we've hired from the operating budget to actually manage these centers. So as you can imagine, the schedulers were used to taking care of scheduling patients who have cancer. Now there's a whole arm of the institute that's actually taking care of patients, getting them in, navigating through the tests that they need, scheduling, et cetera. And then there's also a whole research arm that's devoted to getting those patients who are at increased risk enrolled in all the research studies that we have ongoing for the future. Next slide. And since the launch in 2023, we've taken it step by step. So we can't, we realized at the beginning that we couldn't really do everything on day one. We started with the multi-cancer early detection clinic, which Betsy will tell you a lot more about in the second half. But in the solid tumors, we focused on the people with GI precursors. So thinking of people with Barrett's esophagus, you may know that term, people with cysts in the pancreas. We know that some of those patients, some of them, not most of them, will develop pancreatic cancer, et cetera, and gynecologic tumors. So people who are at elevated risk of developing ovarian cancer or endometrial cancer. Some of them may have a family history of those cancers. Some of them may carry a BRCA mutation. And we have a multidisciplinary program that takes care of those patients over a spectrum, over a long period of time. About a year later, we launched oral medicine, the study of oral medicine, and there are people with oral lesions that have a very high risk of developing squamous cell cancer. So again, we're having a clinic that's taking care of patients when they're having a precursor, so a precursor to cancer, and developing trials and studies based on cancer prevention and this interception concept. And again, so on and so forth. So we now have a study of cutaneous, a pediatric program, and a genitourinary program. And we plan to actually develop these types of clinic in every tumor spectrum. So you'll see breast cancer is not on there, and lung cancer is not there. There are programs that cater to those patients, but we really want to organize this effort to think strategically across the research and clinical spectrum. Next slide. And one of the anchors of the program is something that's called the In Advance study. And it's basically, in order to understand what the factors are that predict who's going to get cancer, what are the risk factors, we really need to get data. And we need, we're really reliant on patients' participation, and we've had a tremendous involvement from our patient population into enrolling in this study, which every patient who comes through one of these clinics gets asked to enroll, and we collect detailed data from them about their behavior. So what are they doing for smoking? What are they doing in terms of weight control? What are their demographic factors? But we're also linking those data to their medical records, to their family history, and very importantly, we're collecting biospecimens. So for every patient, they're getting asked to provide blood samples, but also urine samples, stool for microbiome, tissue biopsies if it's relevant for them. So I think this is one of the pillars, is that over a spectrum of time, that'll give us the capacity to say, what is there in your blood, for example, that could predict whether somebody's going to have progressive disease or not. Next slide. And this is the platform. So for anybody who's listening in and thinks, oh, this is interesting and I may have a risk factor, you actually do not need to be a patient at Dana-Farber. We can enroll you online. Everything is done online. We can send somebody to collect blood at your home. And it's a very user-friendly platform where you can do the consenting online, fill out all the surveys online. We're trying to get an app together so you can do it on your phone as well. And so anybody who's interested, please reach out to us afterwards if you feel like you fit into one of these groups that I've talked about. Next slide. So I'm going to give you a broad kind of overview of the kinds of research. We're going to focus mostly today on the multi-cancer early detection test because we know that a lot of people have interest in the Gallery test or the GRAIL test. But in this kind of same vein of what's the kind of work that we're doing in terms of cancer risk and interception, I'm going to give you just an overview. We're not going to go into the details of these slides. But the first one is something that we've developed in my laboratory called the Prem Plus tool. which is a family history tool that, again, is patient-driven. Patients can fill out their own personal and family history and figure out whether they meet the criteria to get genetic testing. That's specifically for an inherited predisposition to cancer. And we've been working on this for about 20 years. We started in a very specific area of colorectal cancer and endometrial cancer in a syndrome called Lynch syndrome. But the most recent version of this tool is actually looking at inherited predisposition for a wide variety of genes, including BRCA1 and 2. And our work here is actually to integrate this tool into the electronic medical record and the intake forms of patients around the country in large healthcare systems. So everybody is getting a family history assessment and then recommendations regarding whether they would benefit from genetic testing or not. I'm going to skip to the third bullet, EGFR screening. So Dr. NJ Flores is a pulmonologist, a lung oncologist in our group, and she's looking at a blood test based on a gene called EGFR, looking at lung cancer screening among healthy East Asian and Latino individuals because they have a higher rate of lung cancer and a higher rate of this marker. And again, that's looking at sort of combining EGFR screening with what we already do in terms of CT scanning for high-risk populations. And then another investigator is looking at something called microRNAs, another very hot biomarker in the scientific arena, as an early detection test for ovarian cancer. And I'm sure many of you know that, unfortunately, we still don't have a great test for ovarian cancer screening. So the microRNA is a really promising tool that may help in this arena. Next slide. Now, how about early detection? So you're going to hear about the Grail-Gallery test from Dr. O'Donnell. And those are the last three bullets in here, but there are other approaches. So AI for early detection is a big thing, right? So can we use artificial intelligence approaches, whether it's to use that approach to sort of do a deep dive into the electronic medical record to find people's risk and figure out who needs to be followed. But the other piece that's really promising is using AI to do a better job at using our imaging. So a better job at doing mammography for breast cancer screening. And we're doing a project on looking at better AI imaging for CTs for early detection of colorectal, gastric, and pancreatic cancer. There's a lot of technology coming down the pipeline. So can we use novel technologies rather than doing a colonoscopy? That's another project that we have, working with an engineer to develop a capsule that you could actually use as an alternative to colonoscopy. So we're very early stages in that, but I think that wouldn't that be exciting if everybody didn't need to get a colonoscopy and could do something much easier to do. And then again, similarly, can we use stool testing? Can we look at DNA mutations in your stool? We have heard of Cologuard, but we're using Cologuard now in higher risk populations where it's not recommended currently because there weren't enough data. So we're part of multiple studies in this arena of looking at high risk populations and whether stool testing could also be an alternative to colonoscopy. Next slide. And then finally, the interception piece. So I talked about the high-risk oral precancerous lesions. They're a set of patients who have these oral lesions that have really high rates of developing squamous cell carcinoma. So Dr. Glenn Hanna, who is the lead of our oral medicine program in SETI, is actually just launched a trial looking at immunotherapy look at attacking those precancerous stages. And it's remarkable the kind of responses that we've seen. So again, the idea is, can we intervene at that precancerous stage to prevent the progression into squamous cell carcinoma? And then there's surgical approaches. So we know that in BRCA carriers, the standard recommendation has been to remove the fallopian tubes and both ovaries. Now that's a big intervention. And there's an ongoing trial to say, can you actually just remove the tubes and retain the ovaries for a longer period of time so patients can benefit from their hormones that they need till a later date? So this is, again, a large center trial to look at the best surgical approach in patients who are at high risk for ovarian cancer. And then finally, the other big category is vaccines. So you know that our immune system is very involved in immunotherapy. Well, can we use those same approaches to do immune prevention? So Dr. Hanna's study is an example of that, but there's actually the development of vaccines that are targeting the biological sort of the biology that we understand. And there are two vaccine trials that are ongoing in a group of patients known as Lynch syndrome that are in the early stages, and there are vaccines in development for other cancers such as pancreatic cancer. The next slide. And then the next steps. I think one of the big things that we want to make sure is that people have access to early detection and interception strategies, not only at sort of tertiary care centers like Dana-Farber, but that they're available to the community. So a big part of the mission is to look at our satellite sites around Massachusetts and open all these research studies and the approaches like the biobanking that I talked about in our surrounding communities as well. Um, we're really out there engaged in community events. So I think one of the big things that we know for certain things like lung cancer screening, we know that CT scanning works for people who are long-term smokers. Unfortunately, if you look at compliance rates, less than 30% of people are getting that CT scanning that they should be getting. So we have an entire branch of our program that's actually getting out in the community, educating community providers as well as patients to ask for the screening that they need. And then this is going international. So I think being the leader in this has led to a lot of external collaborations. And now we're part of a consortium called ACE, which is the Alliance for Cancer Early Detection, that includes centers in the UK, in Germany, in Oregon, in Stanford, that are going to work collaboratively so we can actually bring all of these studies to even a broader population. And then I think this is my final slide. I think one of the big things is that we need to educate the physician community as well. So a lot of our patients tell us that often they have to be their own advocates about prevention and early detection approaches, especially if they're high risk, because it's often important to do more for those patients, right? So they have to be the ones going to their doctor. So I think one of the big things that we think is really important that we start early at educating the primary care physicians because they are going to be the drivers of this work and the drivers of this care. So we're very excited that we launched our first continuing medical education course. So Betsy and I were partners in this, but every person who actually contributes to our SETI program gave a talk, and we had tremendous engagement from physicians in our local Boston community, but also around the country and around the world who joined in virtually. So it was a really exciting day, but it's really part again of our mission central approach of saying we really need to get the word out there of all the novel approaches that are coming down the pipeline, what's ready for prime time, what's not, and how people can actually be advocates for early detection and interception. So that's my half of this. I'm going to hand off to Dr. Betsy O'Donnell, who's going to tell you about the multi-cancer early detection clinic that she runs and the work that they're doing.
Well, thank you so much for this handoff. And really, I think it's worth saying this has been a labor of love for Dr. Singal and for me. She's been at Dana-Farber longer than I, but I started at Dana-Farber in 2001 as a clinical research coordinator and really was inspired by the mission of Dana-Farber and have had the opportunity over 2 decades to watch as our therapies have evolved and as we've gotten better at treating cancer and all the work and all the resources that go into clinical trials of novel therapies. But I think that those of us who work in this center and really give Dana-Farber so much credit for recognizing that part of curing more people is finding cancers earlier. As Dr. Singal said, when we find cancers when they are stage 1 and stage 2, so when they're localized and haven't spread, that's when we can achieve our highest cure rates. So the question that we have asked ourselves here at Dana-Farber and specifically within this group is how do we take the same approach that we've taken to therapeutics and apply it to early detection and interception? And I think that cancer screening is one of the low-hanging fruits. When we look at the current state of cancer screening and what our guidelines are from the U.S. Preventive Task Force Service, there are only 4 cancers for which we currently recommend screening: for breast cancer, for colorectal cancer, for cervical cancer, and for lung cancer in smokers. What that means is that over 70% of the cancers that people get don't have current screening guidelines. And so to me, that's a low-hanging fruit. How can we start to get at finding more cancers early so that we can cure more cancers? And so one of the things that we'll talk about in a lot of detail today are multi-cancer early detection tests. So let's talk about what these are. So basically, in the last 2 decades, we have developed the science where we can sequence the human genome. Not only can we do that, we can also sequence abnormal DNA. So we know what normal looks like and abnormal. We can look in the blood and find pieces of abnormal DNA that may be signatures of specific types of cancer. So we've sequenced what breast cancer looks like, what colorectal cancer looks like, and there are libraries of these DNA references that we can look for in the blood. And then different companies are now developing tests that they can do at relatively decent speed, couple weeks, and at debatable cost to look for signatures of cancer in the blood. Why is this exciting? Why is this different? And what is the potential? There are no FDA-approved blood-based screening tests at this point. For the remainder of this talk, we'll talk of them as MCED tests, multi-cancer early detection tests. Right now, these are an evolving field. They are called lab-developed tests, or LDTs. Some of the tests that are available for people to buy, commercially available, have been studied in the types of studies that we would do, what we call prospective clinical trials, where we introduce these tests in specific populations of patients to see how well they work. But many of them are still really tests that have been developed by labs and are going to direct to consumer. But the promise of these tests is that they can look for fragments of DNA, depending on the technology, and try to identify abnormal, potentially cancerous DNA from normal DNA. Why is this exciting? Well, it offers the potential of using a simple blood draw to potentially screen for multiple cancers. One of the challenges in trying to screen for so many different types of cancer is the prevalence of each cancer. When you have uncommon cancers, the number needed to screen, so the number of healthy people you would need to screen to find one case of cancer, is very high. And so if you compound that by the number of cancers, it would be very hard for a single primary care doctor to screen for all of the different cancers doing multiple different tests. So if we can put them into one test, we increase the yield and lower the number needed to screen to find one cancer. In addition to that, I think most people are amenable to blood draw as something they're willing to have. It doesn't, require a procedure. It doesn't require taking, large amounts of time to get done. The goal of these is to expand the number of cancers we screen for, so that in a potential test, we can screen for as many as, for example, we'll talk about one test that can screen for up to 50 different types of cancers. The holy grail, though, would be to find cancers earlier. So if you have DNA of a potential cancer in the blood, that means that there are cells breaking down and shedding their DNA. So there has to be a certain burden of disease for that amount of DNA to be detectable. So how do we get tests that are sensitive enough to find even small little particles of DNA to find these cancers at their earliest stages? One of the things that I'm particularly passionate about is expanding access to screening. If you think about, and if you've done a colonoscopy before, you know that that is really a 2-day enterprise, one for the prep and then one for going for the procedure. And it requires having someone to take you for the procedure if you're having sedation. That can be really challenging, particularly for people who, have work at an hourly wage job. So how do we think about screening techniques that might broaden the range but not add to the burden of patients being screened? And so one of the tests that is available to patients is Grail's Gallery test. There's another test that just came on market made by Exact Sciences called CancerGuard. Both of these tests have been studied in clinical trials. The Grail test was just recently updated their results in October at one of our major conferences. And one of the questions that comes up about these tests is, what is their positive predictive value and what are their sensitivity? How well can they pick up cancers? And so when you think about, if you have a test, if each of us were to do a test, about 1% of people who do these blood-based screening tests will have a positive result. That does not mean that someone has cancer. A positive signal implies that you need an additional workup. And so if you have this test, for example, their data show that about 61.6% of the time that the test is positive, there is truly a cancer there. So said differently, if you have 10 people with a positive signal, 6 of them will actually have cancer. That's a really important number when we think about feasibility and doing these tests on a broad scale. What we don't want to do is screen people, create false positive results that increase distress, and also create a diagnostic odyssey where patients have to have multiple different tests or even procedures to make sure they don't have cancer. The negative predictive value of this test, meaning the ability to say if you have a negative test, you don't have cancer, is about 99.1%. And so what is the current state of multi-cancer early detection testing? These are tests, there are at least two now. There are others that use different techniques, but there are right now two that are commercially available, meaning you as a patient could buy them. You could either get them through a primary care doctor, through some type of specialist, or direct from the company through their web-based providers. But that's a very different paradigm than anything we do in medicine. When we give drugs, when Dr. Single and I give drugs, these are drugs that have been studied in very specific fashion in a sequence of clinical trials that culminate in FDA approval. And so at Dana-Farber, we take a little bit more cautious approach to recommending these tests. We're very interested, just like we are in studying new therapies, in studying these new tests and understanding in a general population, especially because there's cost associated with them, if they're safe, if they're feasible, meaning if you get a positive result, can we work these tests up, make sure that insurance covers it? And what do we do if you're one of the false positives? How do we properly make sure that we've adjudicated the test correctly and that we have a surveillance plan in place? So I mentioned how people are getting it, and I do want to talk about clinical trials because we have several, but what happens if a patient has a test? What are the clinical services that we provide to individuals? So if someone has a test through a primary care doctor or through one of the commercial websites, if they have a positive test, we will see those patients. It is imperative to us that we make sure anybody who has a potential cancer is properly evaluated, that we determine if a patient has cancer, and if they don't, if patients have a false positive, that we do all the appropriate tests so that we can give those patients peace of mind that we've properly evaluated them, and then we're going to keep a close eye on them to make sure a cancer doesn't evolve. So if a patient comes in, only about 1% of the general population that gets tested will have a positive test, but we will follow patients to diagnostic resolution. In the past year, so we opened in December of 2023. So in the first year, this goes through January of last year, we saw 14 positive tests. These were not patients we tested. These were patients who self-referred into Dana-Farber. 11 of the 14 patients we saw did in fact have cancer. To give you a sense of the number of tests that it took to resolve this, it was about 4 to 5. For patients to get to Dana-Farber, you can see the lag. So time to resolution, so time from having the test to getting a conclusive result was 48 days. But once patients got to us, we were able to move things along very swiftly, 15 days. But what this highlights is that if you're having these tests and you don't have a dedicated provider, if they haven't ordered it, they may not be able to comfortably evaluate. The way these tests work is they give you a positive signal, so saying that there's a signal, yes or no, detected, and then it will designate a cancer signal origin, a CSO, of where they believe the abnormality, what organ it's coming from. So let's say it's lung cancer. It would say positive screening result, suggestion of lung cancer. And so the onus then is on the doctor to do an evaluation for lung cancer, either making that diagnosis or making sure that we've ruled out and within lung cancer within our greatest abilities. What's also interesting about these tests is they have the ability to screen for both solid tumors, so things like breast cancer, lung cancer, gastric cancer, and hematologic malignancies, multiple myeloma and lymphoma. And what we saw in our experience was an even distribution between the two. One of the really important things we talk about, though, is finding things early. And you can see that more than half of the cancers that we found in our experience were stages 1 or 2 when they are still most curable. What was really interesting is that 90% of the cancers that we found in our clinic did not have recommended screening tests. So coming back to what we talk about, where there's this need for broader screening. Interestingly, we did have one case of breast cancer. It was a triple negative breast cancer in a patient who had had her mammogram, and the mammogram was read as abnormal but thought to be benign. And so because of this positive test, we did additional testing and ultimately did make a breast cancer diagnosis. when we think about where people are having these tests and how they're coming to Dana-Farber, where do these stand in the marketplace? Only a minority of people are actually getting these tests from their doctors. Most people are getting them either from their insurers or from their employers. And most people are coming to Dana-Farber because they've Googled us to find help to get these properly evaluated. So I think the really important take home here is that if you're thinking about doing one of these tests, try to do it through a provider who understands the properties of these tests and is comfortable doing an evaluation if you have a positive result. We believe in studying these tests. We are very fortunate to be able to offer several clinical trials involving MCED tests. The first of these is called the INFORM study, where we are offering free testing to patients who are at high risk for cancer. One of the important things that we're trying to do is understand who best might benefit from having a multi-cancer early detection test. So we're offering free screening to 500 patients who have what are called germline predisposition syndromes, where patients who have inherited risk factors for cancer, and also individuals with strong family histories. Strong family history is sufficient to warrant germline or genetics testing. And so if there are individuals who are interested in that, they can come to Dana-Farber and receive free Grail gallery testing. Just for a reference point, on the commercial space, these tests cost about $950 out of pocket. We also are fortunate to have a Department of Defense grant where we are screening veterans, 1,500 veterans. So if there are veterans on this call who served more than 4 years, we're in the process of amending our protocol from 8 years of service down to 4 years of service over the age of 45. This is a study in which we're not only funding the cost of the test, but if you live outside of Boston, we can bring the test to you. We can leverage the same resources that Dr. Single was talking about through Exam One or Quest. You can either go to a testing place locally or have the test come to your home to offer patient screening. And if you have a positive test, the grant will fund the transportation and workup of that positive test. So if you or a family member or someone you know is a veteran, we will, in addition to looking at 1,500 veterans, we will compare our outcomes to individuals who are seen at the VA as a synthetic control to understand the value of these tests in the population. I think we're really still at the place where we're learning about these tests. They have tremendous potential. They're extremely exciting. We believe they may be able to help us cure a larger percent of the population, but they are still very much something that we are studying about. So the conclusions about MCED tests is they are very much making their way into clinical practice. You or people you know may be having these tests done. It is critically important, though, that if you have a test that has a positive result, that it be properly evaluated. And, there's so much more that's going on. This is an area that is rife with research and extremely exciting. We want to see these tests take the same pathways that drugs have, though, so we ensure that they're safe for patients. This is something that is going on a legislative level. There's a bipartisan bills going on right now. There are parallel bills going on right now with bipartisan support, such that if these MCED tests gain FDA approval, that there will be pathways for healthcare reimbursement to pay for the cost of these tests, which is critically important just in terms of the feasibility of individuals being able to afford these on a regular basis, and also in terms of broadening access so that everybody has the opportunity to benefit from enhanced screening. So we are really thrilled that we are in a position where we're able to offer novel early detection and intervention opportunities at Dana-Farber, and it is an exciting area of research, and I'm happy to answer any questions you have about these evolving technologies. Thank you.
Wow, thank you so much, both of you. You could probably turn the slides off and we can
Sure.
welcome questions from the audience. I have a couple to get started, but I have to say, as a cardiologist, I am so amazed by the changes that have taken place in oncology in the last couple decades. It's so different than when we were
I
training.
know.
Because it used to be like, why screen if you're just going to find something that we don't have a treatment for? And I think many people in the audience who grew up in that era too aren't necessarily, it's not on their radar to go get a lot of tests for screening, because we didn't all know that some of these things are now curable or at least really manageable. So thank you. And I think this whole community outreach and educating the patients and the primary care doctors about this is so important. Um, Now, I do have a question. So a lot of people are now going out on their own and getting these MCED tests, the multi-cancer early detection tests. And for a period of time, that really gives you some peace of mind if it's negative. How often do you recommend that people repeat those? What's the warranty on these tests?
So that's a great question. So there have only been two studies done prospectively in patients, and each of them has been one-time only testing with 12 months of follow-up. And so what the company does is it leverages its real-world experience to recommend annual testing. There is an ongoing study that involves annual testing for 3 years. And I think that will be very informative of the utility of doing annual testing. Because if we look at screening, we don't do, for certainly for breast cancer, we do annual screening, but for colonoscopy, that's not something we do necessarily on an annual basis. And not to be cynical, but where companies stand to profit for tests that are fee for service, it makes sense that they may be recommended more frequently, but we don't have data at this point that support the appropriate interval for testing.
Great. Because I imagine the more advanced a cancer is, the more it's shedding these
That's
DNA
right.
fragments into the bloodstream. So if you were to pick up a stage 3 or 4 cancer, um, is there also like a quantitative response on the test that's really positive or it's a little positive?
No, that's a great question. So it's a threshold level that the company that's proprietary for the company that they set, where it's over a certain amount, flags it as positive.
I see. Okay. All right. And it'll tell you, looks like it's coming from the
That's
colon
exactly
or
right.
the breast.
So the test provides, and not every test is the same. So these are the two tests that are commercially available that actually have prospective data where they've been studied in humans. There are a lot of other modalities and types of technology that are evolving. There's no clear winner yet.
Mm.
But so, when we look at one test, it provides a yes, no plus a signal origin. The other test is just binary, says yes or no, go get a CAT scan.
Oh,
So, depending on which test you have, the workup that's recommended differs. So again, it's really important when you're obtaining these tests, since these do not necessarily go through the conventional channels that we're accustomed to know the properties of the test and know who you're going to call if you have a positive result. We're
mm.
happy to see patients, but if it's not convenient to come to Dana-Farber, then I think when you're ordering this test, you need to have a plan in place for how you can get help.
Okay. So the Grail Gallery is the one that you're talking about most, and that's the one you're all using.
Mhm.
But you're saying there's another one that's
The
just,
Exact Science has what's called Cancer Guard, and that became commercially available on October 1st. That was studied in a study called the Detect A study, which actually involved it was all women. And so it was a different population. They have other studies that are going on now looking at in a general population, both male and female, but the original study was women only, and they used a PET scan to determine whether or not patients had cancer if they had a positive signal.
wow. Okay. There,
And there are
we also,
multiple companies, I think
Right.
besides these two, most diagnostic companies have tests kind of in the development phase. And the issue is that you need to do very large studies, to be able to show whether these tests actually do benefit, because most people are not going to have cancer. So there's, it's more of a concept of, can we use blood testing
I
to
see.
pick up multiple cancers? That's kind of where we're at, and I think that's what Betsy's like, incredibly thoughtful about this, because we don't want, we're firm believers in the approach of early cancer detection, but we also don't want to, because people always say, and we didn't talk about this, like the whole body MRI is the other parallel, right?
Yep.
Seems like a good
Yeah.
idea. Should I get it? Well, we don't have the data, and there are harms to picking up lots of signal that ends up being nothing. It leads to worry, it leads to biopsies, it leads to days off work. So it's really, really important to say this is a great idea, but we really need to figure out, if we're giving people information that actually benefits them and prolongs, improves them on based on compared to doing nothing.
That's
No,
right.
totally. I have so many patients who've had an MRI of the abdomen for some reason and have been found to have these little pancreatic IPM,
IPMNs, right?
right?
I
And now
think
they
it's,
have to get this every
yeah,
year. So that's what you're talking about. Like if you do an MRI of the whole body, you'll pick up things that may or may not be important that you're compelled now to follow up on.
Right. And I think that, but that's, again, the IPMNs, that's one of the clinics we set up exactly for this problem, is that there's so many people who are just being found to have IPMNs. Now, that doesn't mean that, we shouldn't have found them, but I think that they do need to be followed. So, and a lot of people are out there in the community kind of not knowing where to land with that.
Well, what about going in the opposite direction and saying, okay, you've got an MRI that shows an IPMN, let's do the Gallery test and see if pancreatic cancer comes up? Because you're saying 99% of the time, if it is cancer, it'll pick it up. Would an IPMN be negative? And therefore, could you say we're done?
So that would be a great study. Something that hasn't been done is what is the sensitivity and how can we better predict if, one of the things that you think about, I mentioned a patient who had triple negative breast cancer. Their test is better at picking up the triple negatives because they tend to have more turnover. Even though hers was an early stage cancer, the cell turnover is higher, so there was more DNA. So you could make a conceptual argument that something that was more proliferative and had a greater malignant potential might be picked up. We do have a study. It's not exactly like this. I didn't put all of our studies up. This one is IRB approved, but not yet open, where we're studying these tests for cancer diagnostics. So I also run our cancer diagnostic program at Dana-Farber, and we see patients who have radiographic image findings concerning for malignancies. And, when you're doing a cancer evaluation, sometimes we have non-diagnostic results. Sometimes that lymph node is really hard to get into through invasive procedures. So we're actually studying whether we can use these tests for helping to aid the cancer diagnostic workup. And I think that really aligns with what you're saying too, is, can we also pick out maybe some of the more aggressive things that are on the spectrum of potential malignancy by these tests? I don't think we have an answer to that yet.
Interesting.
But I think it's important actually, Dara, to address one other point is that one of the risks of these tests is false reassurance, right? So, in like the IPMN question, we don't know, as Betsy's saying, whether
Right.
those tests actually work in the IPMN population. But if somebody with an IPMN goes out and says, "Oh, I've had the Gallery test, it was negative, I don't have to be followed,"
Mm.
that's like a risk, right? Because then they've gotten falsely reassured. So again, the MSAT tests are going to be complementary.
Right.
It's not going to get rid of a mammography. It's not going to get rid of colonoscopy because we have reams of data. And so it's additive, exactly as Betsy said, for those cancers that we actually don't have great screening tests for. So that's an important point, I think.
Yeah, I want to underscore that these are not at all meant to replace screening tests that are
Mm.
the four that I talked about remain the gold standard, and these are really under investigation as a complement to those to broaden the range of screening. One of the things I do want to say is, the other hat that I wear is I'm a medical oncologist and I work in plasma cell disorders, multiple myeloma, and the precursors. And what you said too is so true that when we didn't have good therapies, why would we find things earlier? And so when we can identify precursor conditions, whether they be IPMNs, Barrett's esophagus, MGUS, and smoldering myeloma, we can follow people over time and we can learn about the biology. And one of the things that we do is spend a lot of our time and research on is trying to, if we do find someone early, better understand who will be the progressor versus who will not. Who do we have to worry about
Mm.
versus who we don't? If we don't study those patients, we don't answer those questions. And so I think that's really an important part of what Dana-Farber is investing in terms of early detection. It's understanding risk of progression as well.
Got it. So interesting. I mean, can you say a word? I know neither of you are prostate cancer people, but since it is such a common cancer, is there also a role there? Because in terms of these very slow growing cancers that a lot of people would say, oh, you have this, it's not likely to ever kill you. Everyone has prostate cancer after the age of 85 or whatever. Does this test help determine a slow growing, not benign, but less malignant cancer that's not likely to cause a problem versus one that we really need to jump on quickly?
No, it doesn't have that. And unfortunately, one of the tests this does not do well is prostate cancer. So the prostate is an encapsulated organ. So unless the disease is advanced, it's unlikely to shed. If it were positive, though, so if you did get a positive prostate signal, then it would heighten your concern for that malignancy.
Okay.
But no, this test doesn't have the ability to differentiate between doesn't in terms of aggressiveness or risk.
Okay. Um, Great. So once it gets into the bloodstream in prostate cancer, at least, that means it's sort of.
And this technologies, Dr. Singal alluded to this. This is what's currently available. I will often say it's a little bit like the early days of the iPhone, or even when we were using Blackberries, where these are all exciting new technologies, but they're going to evolve, and some of them are going to keep evolving, and others are going to go, by the wayside. And so there are a lot of other things besides circulating DNA that we're looking at. Things like your immune system signals. As someone is fighting a cancer, there are changes that occur in the immune markers, their B and T cell repertoire. So that's just one example where people are studying those changes and trying to come up with tests that they could maybe pair with these DNA tests, or you even use as a standalone, looking at the immune response as a marker of cancer rather than specifically for circulating DNA.
Oh, that's so interesting. Okay, so this understanding of cancer has also evolved so much, and the immunotherapies that you were alluding to um, relate also to the fact uh, that the immune system is starting to react to the cancers as well.
Mhm.
Can I ask you a question? You were talking, this is a little off topic, but talking about vaccines as part of the interception for certain types of cancer. Does a vaccine treating or preventing a type of cancer imply that this is related to an infection, or how is that? How do we think about vaccines and cancer? What's their
Yeah,
relation?
I think it's a great question. So, the reason why you're thinking about infection is one of the big home runs of cancer prevention has been cervical cancer, right? And both due to screening as well as vaccination. So the cervical cancer vaccine was built around the viral HPV against a virus. The other immunoprevention approaches are actually not there, but the biological aspects that are not always infectious.
Mm,
So for Lynch syndrome, like I was talking about, is anything that will kind of stimulate your immune system to kind of overact and attack the precursors or the cancer, the cancer sort of the pieces in the development. So it doesn't necessarily have to be infectious at all.
I see.
So the
Okay.
pancreas vaccines, for example, are targeting KRAS. So that you can actually do against KRAS and as an approach for pancreatic cancer. In Lynch syndrome, it's looking at microsatellites, et cetera.
Sorry, can you explain what KRAS is?
KRAS is a gene that's involved in pancreatic cancer development.
Got it. So, okay. So again, not exactly what you were talking about before, but if someone were at high risk for pancreatic cancer because of a family history or other things,
But yep,
you're looking at potentially offering a vaccine.
vaccinating, right.
Oh,
And I think part of what you said at the very beginning, Dara, is that really important, I think, is that we want to, people are traditionally scared about getting cancer and scared about cancer. And I think that I'm amazed that I didn't use this analogy in this crowd because I always do, but I'm going to use it now, which is that we would never wait for a heart attack, right, to check somebody's blood pressure and cholesterol. And yet we do that for cancer. And this is the whole thing. We want to change people's thinking about all these risk factors as being your blood pressure and your cholesterol with this proactive approach and saying, look, we can actually intervene and actually have an impact on cancer. And I think that's the messaging that we need to kind of shift around the most is people to feel like this is something that's empowering rather than scary. And we have the tools to do it. So the IPMNs, the MSED tests, but we really need to kind of organize around that question, if that makes sense.
that makes so much sense. No, and I love that you use the word interception instead of prevention because you're sort of interrupting it, right? Something
Yes.
that could otherwise be almost inevitable. Are there any tips you'd give to the general population who may or may not be at particularly high risk? Are there general preventive measures we can all take that might lower our risk of getting cancer in the first place?
Want to go Betsy first?
Sure. So, there are certain healthy lifestyle advantages that we can help ourselves, staying normal weight, exercising regularly. Exercise is the best medicine. What does the American College of Sports Medicine, the American Cancer Society recommend? They recommend 150 minutes of moderate intensity exercise per week, doing strength exercises that work the core muscles, 2 to 3 sets of repetitions, 2 to 3 days per week. These are some fundamental things. Limiting alcohol consumption. Last year, the Surgeon General officially, we've known this, but named alcohol as a carcinogen. And the current recommendations are that women not drink more than 1 drink per day and men not more than two. Trying to get sleep. So, an adult should sleep a minimum of 7 hours per night. That can be challenging, but these are just some of the general guidances that are recommended. Obviously, trying to avoid tobacco, smoking tobacco is another important consideration, and mitigating stress, and trying to have relationships are actually important for health too, and welfare and mental health. So having positive relationships in your life is also, these are kind of the core values of lifestyle that can help patients. We're seeing so much change in the lifestyle space, and that's one of the things that we study as well, is how in the early detection interception phase, where we know people have conditions that have a risk for moving towards cancer, are there things that we can do, whether it be prolonged overnight fasting, use of the new GLP-1 drugs as early interception targets for patients at risk? And I'm going to pass it off to Sapna to talk about family history and risk.
And I was going to say that, and the other big thing is know your family history. I think, I mean, that's, I think we think now of this whole thing as precision prevention, meaning like it's a targeted approach to prevention, right? So at 2025, everybody really should know their family history. And I think if that's one of the big things, so Thanksgiving is coming up. The Surgeon General of like a previous generation has sort of dedicated Thanksgiving to be Family History Day. And I think it's a great time because one of the biggest things is that people don't know cancer diagnoses. They don't know where it started. They don't know the age of the diagnosis. They don't know the specific organs. So I think to keep like a catalog of what your family history is really is important in today's days because we're being much more precise in
Mm.
our approaches to prevention. And you may be eligible for breast MRI if you have a strong family history and if your risk sort of meets that threshold. So I think starting the conversation early and getting genetic testing, if you meet the criteria for genetic testing and do it early, because I think, again, the key is to start thinking about prevention, early detection earlier in life than what we're used to, you know.
And I think
We
that's
talk about red meat. That's the other for
oh,
June.
that's another one. No
Red
more
meat.
red meat
Red
once per
meat.
week.
Yeah.
Try to limit sugar and anything white. If you can't, shoot it or pull it out of the ground, it's probably not great for you. So try and do 2/3 of your diet. Fruits and vegetables is always, better if you can do that. Also, one other thing too on family history that I think is so important is right now screening is a one size fits all. We have these, general recommendations, but particularly when you know your family's history, that does change the
Yeah.
recommendations. And Sapna could speak to how it impacts colorectal cancer, breast cancer, and even prostate cancer. So we no longer generally recommend prostate cancer screening. It's meant to be a conversation where you weigh the risks and benefits, excuse me. But if you have a family history, that changes it. You begin screening, knowing if it is recommended to screen. So I do think family history is so critical because it could change your personalized approach to cancer screening.
Thank you. Yeah, Sapna, did you want to add anything to that?
No, I think that covers it. I think it's knowing it and it's really knowing why it's important to know it, right? It's important to know it because it could impact the screening you get, the age that you start, how often you do it.
Yeah.
All those things matter, right? So having the sort of the information at your fingertips and communicating it to your doctor is really important. And I think, one of the things that we're also realizing, and it's really important, is the people on the front line, the cardiologists, the primary care doctors, the gynecologists, the gastroenterologists, like those are the people who actually need to the patients have to have the conversations with, right? So it's really, I think that's one of the big challenges in implementation. It's really important that we get the word out about all these factors and why they're important and people to kind of buy into it. I think we're getting there.
Yeah.
But the multi-cancer, I think that's the other piece, is this whole field is galvanizing to say, let's not stay siloed into the cancers that we're used to thinking about. We really need to think across the spectrum of cancer prevention, early detection as a whole.
Yeah. No, and I love that the analogy to cardiology because I think both fields have evolved so much in the last 20 or so years where things that used to be, we called an LAD lesion a widowmaker, no longer, right?
Right.
People who have heart disease survive for many decades with it, and it's a manageable condition. And in cancer, it's more of an interceptable condition, perhaps. But the risk factors are so similar And I
Mhm.
wonder, some of the common denominator there is reducing inflammation. So keeping your
Mm.
weight healthy, keeping your insulin and sugar levels down. Um, does inflammation play a role in cancer development as well? We know it does in cardiology.
I don't think like, yes and no. Like
Yes
it's
and
like,
no.
like how would you measure? And there's no inflammatory index. But I think again, as we were saying, the immune system definitely plays a role.
Yes.
And I think that this idea of inflammaging is something that is a very kind of interesting idea and hot area of study in oncology. We know that, cancers occur with greater frequency as individuals age.
Yeah.
And then if you compound with that, whether it be obesity mediated inflammation or other toxin induced mediation, then you amplify that. And we don't know what the exact circuits are. It's not well teased out, but I definitely think that there's a piece of that that is very kind of new and evolving within oncology. I think cardiology is probably a little further ahead than we are in understanding the exact kind of physiology of how one gets from point A to point B, but I would say oncology is trying to catch up.
Well, cardiology is a little bit simpler. I mean, we can cut an artery up and look at the layers of plaque. You
Yeah.
guys have like 50 kinds of more cancers. All right. And you had mentioned just in passing that GLP-1 agonists might be something that might help prevent cancer. Is there some early signal to that effect?
So in the field that I work in multiple myeloma, it's one of the known obesity is an associated risk factor for multiple myeloma. And where we have a precursor condition, MGUS and smoldering myeloma, it's something we're studying, looking at the use of tirzepatide, for example, in obese patients who have these precursor diseases so that we can see this is the type of study we're trying to do to get to the evidence that is missing. If we introduce weight loss and if we introduce drugs that can potentially mitigate risk factors that we know are associated with these diseases, can we then attenuate the risk? So we're asking those questions and we are doing those studies.
Wow.
And one of those things, Dara, as you mentioned, in the cardiologies, in the simpler part of cardiology is that the events happen quicker.
Mm,
Yes.
One of the
Mhm.
big
true.
populations with cancer research in this field is that cancer takes a long time to develop. Now, that's like a positive because it gives us lots of time to intervene, but it's a really hard thing for the science because it takes a long time and lots of patients to do the trials, right?
Yeah.
So for the GLP inhibitors, like I think that's like, when do you need to take the drug? How soon? Like, do you need to take them for 10 years? Because we don't, like, so the question is there, but it's going to take a while to answer it. But it seems like since weight loss is an independent predictor, right, of reducing your cancer risk, it would seem that'll work, but it may take us a little while to figure that out.
Yes, without a doubt. Um, One more question before we wrap up. A little bit more, you said red meat no more than once a week. And I know with alcohol, even zero is better than a little bit. Are there other dietary recommendations you could make that have general benefits in terms of cancer risk?
The ones that I mentioned are really the top of the food, just again, avoiding sugar, avoiding white food, so processed food, bleached white food, and really focusing on just getting high fiber intake, whether it be through vegetables or fruits. Those are the critical ones that are kind of uniformly included in our guideline. And I imagine they're very similar, like the healthy plate that's recommended.
Mm,
And this is again the challenge is there's so many different types of cancer and the latency is so long. So some of the guidelines are very similar and unrefined compared to what our curiosity is for knowing specifically what we can and can't do to prevent the occurrence of cancer.
Are there carcinogens in fried foods, like foods that are cooked at very high temperatures, that we need to be worried about? Or is that just colon cancer?
Cured meats, so that's the cured meats are have been shown to be a risk factor really for a lot of GI cancers. I'm not sure of the data in other cancers, but I think processed meats, ultra processed food that Andy Chan, I think, just had a paper that got published in the last couple of weeks that looked at the effect of ultra processed foods on cancer risk and showed there was the higher colorectal cancer risk if you had. So I think it's the same general principles
Okay.
that we're used to. Fresh fruits and vegetables, avoid processed meats, and cured
And
foods, smoked.
grilled meats, also, there's something about that high heat. It's
There
just
is,
Grilled if it's
you know,
charred, but
it's
remember
the
grilled
char.
food is like Mediterranean diet is all grilled. So like don't, throw
I
grilling
think
it on
it's the
its own.
char
It's the char.
specifically, and then also I have read, that boiled, water cooked vegetables versus high heat charred, roasted things may have, better potential. Cruciferous vegetables, things like broccoli, cauliflower. But, these are kind of population, I believe, based data. I'm not as familiar with them.
okay. Cool. Thank you. Okay. Gosh. All right. I could keep you all here for another hour, but I know that we're out of time. So I want to thank both of you so much. You've talked about detection, interception, treatment, all the things that are evolving so quickly in such a short period of time, and then tips for all of us on how to stay healthy and for people who do find themselves either to have a positive gallery or who are at high risk. We'll send out the information on how to get in touch with you all because it sounds like these studies are still ongoing and you're still looking for people. So also veterans, and I think you had firefighter study as well.
Firefighters and high-risk individuals, strong family history. So,
Okay,
and
Yeah.
that is, a lot of people qualify for that study. And there again, we're moving to a decentralized approach too. So don't necessarily need to come to Dana-Farber for testing.
perfect. Okay, I thank you both so much, and thank you to the audience, and hope everyone has a wonderful Thanksgiving
Thank
and
you.
nice
Thank
holidays.
you, Darren.
Thank you.